So the next segment, I'm Kang Shen, so I'm the director of the Tsai Institute. You'll hear a lot from me more towards the end of the symposium. But now the next segment of the symposium is the presentation of the Sammy Kuo Award. And I will welcome Dr. Corinna Darian Smith to present this award. So Corinna is a professor in the Department of Comparative Medicine here at Stanford University, and congratulations to Corinna's promotion. That was last year? Okay, it was since we had a last symposium. So I'll hand it over to Corinna.
Thank you so much. So I get the pleasure every year of, or the last many years, of presenting the Sammy Kuo Award. And it's what I want to, just a couple of housekeeping things to begin with. There are some of you who will be receiving an award who aren't here today. That's fine. I've got mugshots of all of you. Those of you who are here physically, I'll have you please come up as I introduce you and your work, and Zulima will give you your award at the side here. And then I'll ask you to stay and so that we can have photographs later on. So each year I like to give a little bit of a background spiel about where the beginnings of this award came from. Sammy Kuo was born in Taipei, Taiwan in 1984 to parents Yao Shi and Li-Ling Kuo. And in 1996, Sammy was just 12 years old, and he was diagnosed with a rare inflammatory disease which is associated with the measles virus. And his parents looked after him and cared for him at home until he passed away in 2006 at the age of 22. After his passing, his cousins Peggy and Amy Chen, who were both Stanford graduates at the time, and I believe that they are actually watching this live stream, so welcome to everyone in the family who may be doing that. They contacted the Neurosciences Institute at the time, and the Sammy Kuo Memorial Scholarship Fund was established to support basic research in neuroscience. The opportunity allowed the family to memorialize Sammy, and it recognizes the crucial importance of neuroscience research that may one day help cure other children with debilitating conditions. And this award has become truly prestigious, and though born of sad beginnings, it's a real celebration of spectacular work, and it highlights and recognizes exceptional early career papers that have been published within the previous year. It's now in its 15th year, and if you have interest, and I would recommend everyone go to the Sammy Kuo website and you'll see a lot more information. But it starts to read, the first prize getters start to read as a who's who in neuroscience, certainly at Stanford. Before we begin, I just want to give thanks to those working behind the scenes. There is both a postdoc selection committee and a grad student selection committee. They don't talk to each other. They operate quite independently from each other, and I want you to keep that in mind going forward. So thank you to everyone on those committees for reading the papers and making decisions, which are often quite difficult. And I also want to give thanks to Zulima and Jill, who really made this possible behind the scenes as well. So let's begin this year with the grad student component of the award, and an honourable mention was made this year to Emily Kubota from Grill Spector Lab. And I'm not sure that Emily is here today. Aha, come up, Emily. And we can congratulate her. Her paper was published in Cerebral Cortex. So Emily used cytoarchitectural data as well as diffusion MRI and functional MRI to examine the relationship between cytoarchitecture, white matter connections, and function in high-level visual areas involved in reading and face recognition from childhood to adulthood and within a large cohort of participants. What they found, and what she found, was that in children, white matter connections are linked to cytoarchitectonic areas rather than being category selective. That is, for example, face and word selective regions in the same cytoarchitectonic area have the same white matter connections, but face and word selective regions in different cytoarchitectonic areas have different white matter connections. This is different in adults, and she found that in adults, white matter connections are linked to both cytoarchitecture and category selectivity, as the face and word selective regions in the same area become more distinct. Emily's findings are important as they suggest that the neural hardware underlying the processing of categorical stimuli are more domain general than was previously thought. And this is especially true in childhood. So very beautiful work. Thank you, Emily. Our third place prize winner in the grad category, graduate student category, goes to Amelie Schaefer. Is Amelie here today? I think she's not here today. Okay. You can see her mugshot anyway in the slide. She comes from the Cole Lab, and this award was given to her for her paper in Frontiers of Physiology. Amelie's paper demonstrates a novel method to correlate 3 known biomarkers of neurodegeneration, and these are all well known: amyloid beta, tau, and tissue atrophy. But the correlation between them has not been understood. The study integrated neuroimaging data over time, physics-based modelling and machine learning, so a wide range of technical skills involved in this, to demonstrate that amyloid beta is a strong predictor of tau pathology and brain tissue atrophy in Alzheimer's disease. Her results for the first time quantify the correlation between these biomarkers, and this now allows for a quantitative, personalised prediction of disease progression, which has not been possible before. So very beautiful work. This year's second place grad award goes to Arvind Virakumar. And you can come up. For his very beautiful paper in Nature. Arvind used single-cell RNA sequencing to characterize 2 distinct cell types in cardiac parasympathetic neurons in the nucleus ambiguous in the brainstem of mice. These cell types were shown to be part of 2 distinct cardiac control circuits. So this was technically very challenging. The first cell type, which is shown up here in light blue in the summary figure, and actually you can point to it if you like, Irene. This one here, the ACV, was shown to be central to the classical cardiovascular control circuit. These neurons innervate parasympathetic neurons on the surface of the heart and mediate the reflex where heart rate is slowed in response to increased arterial blood pressure. The second cell type, and I'd like you to point this out, the dark blue, the ACP cells, these are central to a novel circuit that coordinates heart-lung function, and these neurons act to slow heart rhythm and to drive bronchoconstriction. Individual neurons, which was fairly amazing, bifurcate to both the heart and the lung and are activated in mice by the dive reflex, which requires a coordinated reduction in the heart and lung function to conserve oxygen while the mouse is underwater. Arvind's findings transform our understanding, which is pretty basic at this point, of our understanding of the neural control of the heart and lungs, and provides a new foundation for cardiovascular and cardiopulmonary medicine, and hopefully new therapeutic approaches. Arvind was recently the only student selected to present his work as a hot topic at the International Society for Autonomic Neuroscience in Cairns, Australia, so I hope that was a good trip. Okay, thank you, Arvind. So this year's grad student paper of the year goes to Zhulu Sun from Krishna Shenoy's lab. She unfortunately can't be here today, but it's for her very beautiful paper in Nature. She and her collaborators were interested in the neural basis of a particular form of motor learning. Monkeys were trained to hold onto a robotic device and reach to one of several radially arranged target locations, Once trained, a force was turned on that was orthogonal to the direction of arm movement and proportional to its speed. Initially, the reaches were curved due to the applied force. But over time, the motor system learns to counterbalance this. So after many trials, about 100, the arm trajectory is again straight. Zhilu measured the response of populations of neurons from motor and premotor cortex using many different approaches, V probes, Utah arrays, neuropixels, and she made, with her collaborators, a number of different discoveries, important discoveries. First, when the monkey prepared to make a movement, the neural activity achieved a state consistent with the reach about to be made. Second, when the curl force was turned on for a set of trials, the preparatory state shifted in the direction expected to counteract the applied force field. It did this in two-dimensional space, identified for reaching in two different targets. Surprisingly, though, the neural preparatory activity also moved in a third dimension, as if to indicate that learning was engaged. And the direction of this uniform shift was unique for each curl field and reach direction combination. And finally, during the washout period, when the force field was removed, the first two dimensions of the preparatory activity returned to what they were before learning, but the third dimension did not return, and it appeared to continue to indicate that learning had recently occurred, so a memory. It's very beautiful work and describes an entirely new form of memory. Zhilu is now doing her postdoc at UCSF, and very, very beautiful. So now let's turn to the postdoc category. And Oma Riva, who can't be with us today, Felicity Gore and Kevin Kelly, who I think are here today. So the three of them together received third place for their joint paper in Nature. Nature seems to be becoming a theme in this series. Neural organoids are great for in vitro studies, as we know, of human development, but their lack of in vivo circuitry limits their use in the study of cellular maturation and the integration of human cells into neural circuits. So in this landmark study, Oma, Felicity, and Kevin developed a new platform for transplanting intact human cortical organoids, or HCOs, into the somatosensory cortex of newborn rats to generate a unit of human cells covering about a third of the rat's cortical hemisphere. It's a huge area. They combined this with MRI monitoring of engrafted cells in long-term experiments and made three important discoveries. First, they show that neurons from transplanted human cortical organoids undergo structural and functional maturation, which has not been seen and is not seen in organoids in vitro. This now allows the study of physiologically relevant transcriptional changes in human neurons. Second, the maturation observed in transplanted human cortical organoids makes it possible to uncover new disease phenotypes in human neurons derived from patients. And they showed this in a very elegant example with a rare form of autism called Timothy syndrome, where they transplanted neurons and showed a defect in dendritic complexity, which is not seen in vitro. Third, they showed circuit-level synaptic and functional integration of human neurons into the rat's somatosensory cortex. So their work provides the most comprehensive input-output mapping of transplanted human neurons to date, and it establishes a new model for uncovering circuit-level changes underlying disease phenotypes in neurons derived from human patients in an in vivo context. So a very exciting development in the field. Thank you. This year's second place goes to Daniel O'Shea, also from Krishna's lab, and also on the same paper, for the same paper that Zhilu just won the grad award. Published in Nature, this work required an astonishing combination of techniques and is the first neural population study of classic curl force field learning. So again, this work is groundbreaking in revealing this new form of motor memory, or increasing our understanding of it, and it's very thematic to today's symposium. And if you look up here, you'll see that the monkey is now smiling, because 2 people got this. First place this year goes to Darian. For her very spectacular Nature paper. And she wanted to know if CSF, it's kind of a crazy idea, I have to say, but it was fabulous that it worked. She wanted to know if CSF might regulate brain aging, and whether the transfer of CSF from an older or younger organism, in this case a mouse, or was a rat, mouse, could help identify brain rejuvenating factors. So she developed a CSF collection method, which is incredibly difficult in mice because of the tiny volumes involved, and established an in vivo infusion technique to deliver mouse or human CSF into young and old mice. Amazingly, she showed that young CSF actually does lead to an increase in what are called oligodendrocyte precursor cells, the proliferation of these. These are involved in the creation of myelin around axons in your neurons, and a striking reversal in age-related memory impairment, which is pretty extraordinary. She then identified serum response factor, or SRF, as a mediator of OPC proliferation following exposure to young CSF. She showed that with age, SRF decreases in hippocampal OPCs, but the pathway can be induced with the infusion of young CSF. Tal then screened for potential SRF activators in CSF and found that fibroblast growth factor 17, or FGF17 infusion, is sufficient to induce OPC proliferation and long-term memory consolidation in aged mice. And when FGF17 was blocked, cognition was impaired in young mice. The work demonstrated the rejuvenating powers of young CSF to restore oligodendrocyte function in the aging mouse brain. As you might imagine, this is very much something we all want to hear. You can just, get yourself some CSF from a young whatever, put it in your system and buzz your uncle. No, it's very spectacular. It's received a lot of attention, and it's been covered by, at the time that the application came in, or the submission came in, 144 news outlets, including the New York Times, NPR, and so many others. And you can actually find Tal giving a talk about it online very easily if you just go on and find it. Very exciting work. Thank you very much. Thank you to all our winners this year. And I think it's, lunchtime. I will ask those who have won, people at the front, to stay here for the moment, because we need to get a group mugshot of you before you go away. Okay, thank you, everyone.